talked about TGF beta signaling, then JAK-STAT pathways, then receptor tyrosine kinases. Your TGF beta was receptor serine kinase and others are tyrosine kinases, right. Even JAK-STAT cytokine receptors are also tyrosine phosphorylation. So, what these pathways are doing, they are dependent on, there is some kind of post translational modification which is associated with them, that is mostly phosphorylation which activates the pathway, and the intermediate molecules such as if you take PI3 AKT where AKT comes gets activated, or your STAT risk transcription factors or SMADs, all of them they get activated by the phosphorylation of those molecules and then they transduce the signal, right. This is the general scheme that we have seen. In addition, that we have not, what we know that in those pathways, when these pathways are shut down or when these pathways are inactive, there is no active ligand present, we did not talk about the regulation of the other molecules, how do they get regulated. Let us say what happens to STATs or SMAD proteins, maybe they are just remain in the cell cytoplasm in an inactive state, right. Maybe there will be additional regulatory mechanisms. So, apart from phosphorylation, we have not seen any other major post translational modification which is an integral part of regulation of these signaling pathways. However, there are certain signaling pathways where ubiquitination is an integral part in addition to the phosphorylation, ubiquitination of some of the key molecules. So, SMADs were the transcription factor, STATs were the transcription factors. In this case, AKT is another, it is not a transcription factor, it is a kinase which goes and regulates the downstream molecules, or all the key signal transducing molecules are, they were regulated by another manner. So, there are certain pathways such as Wnt and Hedgehog, or I think Notch as well. These guys here in these two pathways, the key molecules which are going to do all the important, the most important function, like in case of Wnt, you guys mentioned about, spelling is correct, no, beta catenin, and in case of Hedgehog, these are the glia transcription factors. These two factors, the regulation of these two factors in absence of pathway activation or ligand is regulated by a ubiquitination. That is the most important, that is one of the important thing that one should remember when we are going to talk about Wnt and Hedgehog signaling. So, we will get to this in detail, we will talk about it, but do you guys, all of you I think by now have, I mean we have talked about ubiquitination in several times, right, in the class. So, all of you know what ubiquitination is. Do I need to again repeat everything, or you will help me with that? Pratibha, do you want to tell us what do you remember about ubiquitination? Yeah, I remember that. Okay, tell whatever you remember. What do you know? Proteins is mediated by ubiquitination. Good. Anybody else? Rutuja, do you think we can add something to that?
So, there are specific residues of lysine where the tags are added.
Specific residue, specific lysines which are present on ubiquitin, right? Do you want to add something to this, Harshita?
I mean the specific residues where like particular ubiquitination is added determines its fate. It is going to proteasomal degradation, lysosomal.
They form different kind of chains. Most important thing, ubiquitination is a post translational modification, but it is reversible, right? Why? Because it can be removed, and here ubiquitin is a 76 amino acid polypeptide, and like your phosphate, which is just an inorganic, the phosphate we add, no, something, and it does not make a lot of change in the protein as such. Ubiquitination is 76 amino acid ubiquitin is 76 amino acid polypeptide, which is covalently linked to the protein. That is important, and it can be removed, and then it is attached to the lysine, as you all of you guys said. So, there are two things which you should remember, because we did talk about EGFR and then we talked about, I think, ubiquitin. That was the context, right. So, in that case, when we talk, there are two chains, two types of ubiquitin chains that are very important at this point, and there is another pathway when we will talk about other type of ubiquitin chains. But to the least, what you guys should always remember, should remember, is there are two types of chains. K63 and K48, where the ubiquitin will be added onto the ubiquitin. So, if this is one ubiquitin which is added, the next ubiquitin will be added on the 48th lysine which is present of this ubiquitin. The next ubiquitin will be added on the 40th lysine of this ubiquitin. The next ubiquitin will be added on the 40th lysine of this ubiquitin. This is how the polyubiquitin. So, this is, you will say this is a tetraubiquitin, but people call it then polyubiquitination or chain, right. Similarly, you can have K63 like this, and this the reason why I am drawing like this, because these ubiquitin chains also have certain topology to them. It is not random. So, that is not very important at this stage, but remember K63 and K48, and as you know, K48, what will happen to K48 linked proteins? Proteasomal degradation. K63, most likely they will undergo in a lysosomal degradation. This is what very important, and both the pathways, Hedgehog signaling as well as your Wnt signaling pathway, in both of these pathways ubiquitination is integral, because in the absence of ligand, when the pathway is inactive, the key molecule, the key component of the pathway will be degraded, so that you can keep those levels of those molecules very less in the cells. So, that is how you regulate the pathway, that is why it is less. So, very interestingly, so many years ago in 1982, when good. So, many years ago in 1962, okay, 82, this Cell paper was published, where these two people, Royal Nuss and Warmis, I guess, these people, they were doing some interesting studies. What they were trying to do, they took these mouse memory tumor viruses. These are retroviruses. Do you guys understand what is the difference between retrovirus and lentiviruses and other type of viruses, right? So, what they did, they took these viruses, and these are mouse memory tumor virus. So, the name, what does it suggest, that these viruses, whenever you put them into mouse, they might cause the breast cancer, right. This is what they knew. What they wanted to understand in that time is that whether, because this is a virus, and when you infect the cells with these viruses, whether the viral genome will go and get integrated into the mouse genome. That is the question they asked. That is what they wanted to do. So, when they wanted to do this, what they did, they took these viruses, they infected mice with these viruses, and they started looking for it, whether it gets integrated or no. Eventually, they found out that indeed this viral genome gets integrated into the mouse genome. And they did so many experiments, and they found that the integration happens in the mouse genomic loci, which is near this a gene. They identified a space, a locus where it gets integrated, and they named it as a INT1 gene, because it is called as for integration. So, this is the place where it got integrated. Experiment is very simple. What they knew, these are the retroviruses that can cause cancer, right. What they did not know, how it is causing, or whether the genetic material of viruses can get integrated into the genome, mouse genome. That is what they did. They infected and they eventually identified that indeed mouse, this viral genetic material can get integrated into mouse genome. It was not known before that for this particular virus. So, what they and they identified wherever it is getting integrated, that locus, they named it as INT1 gene, and eventually they found that this integration lead to the enhanced expression of other oncogenes. You guys understand the concept of oncogenes versus tumor suppressors? Gaurav, what do we understand by oncogenes versus tumor suppressors?
When oncogene get mutated, it is expression of more expression to lead to cancer.
Very good. And tumor suppressors?
Regulatory genes like Bb628, it will be get mutated, it enable the cell proliferation.
So, what are tumor suppressors?
It means it regulates the cell cycle.
How?
By suppressing the something.
Yeah, so they essentially, those are, you can simply put them as a pro-proliferative and anti-proliferative genes, right? And then people name them tumor suppressors because what happens is that, like for example, you take P10, right? When you have P10 mutation, the cells will overgrow or they will hyper-proliferate, and then it will end up in a formation of tumor. That is why they call it as a tumor suppressor, the presence of this gene. So, what they found out that whenever this MMTV virus gets integrated into mouse genome, whichever, and the locus is this INT1 gene, right. Whenever it gets integrated into this INT1 locus, it leads to the enhanced expression of some of the oncogenes, and eventually it leads to the cancer. This is what they did when in 1982, and these guys were like, well, look, it's a great discovery, right? They identified some mechanism. Interestingly, eventually there were some other people, they were working on a model system called Drosophila melanogaster. All of you guys know what Drosophila is? Have you guys seen any time what is Drosophila? Vaishnav, have you seen any time? Never. Do you eat bananas? So, just keep bananas in your room and let it become completely blackened. Some small in this thing will come, no, flies. Those are Drosophila. These are not housefly. Housefly is bigger, black color one. This Drosophila will be very teeny tiny, chintu ones. Did you guys have watched this movie called Iga? There was a Telugu movie.
Yes.
That housefly one, Rajamouli's old movie. You did not, you don't know. Zoya is like, what is this? Suddenly, why we are talking about that? So, wo wala fly nahi, that is housefly. This is Drosophila, very teeny. Thomas Hunt Morgan, T.H. Morgan is called as the father of experimental genetics, right? He started using
a wind protein
Drosophila
which gets
melanogaster
secreted out,
as,
and it
and
has
apparently
certain
these
roles to
flies
play.
love alcohol.
It
undergoes. So, in
some
fact,
kind of
if you leave
modification, some
anything which is sugary,
post-translational
they will
modification.
go and start doing. That's exactly why on banana you will get is because it might start fermenting, no. Oh, kala banana, it becomes very bad, no. So that's how it is. So you can do this experiment and then you can find. Don't bring them to lab though. So what people, and Drosophila has been used extensively as a model system, right. So what these people do, there were two people, and in 1985 apparently there were two of these scientists. I do not know if I missed their name. One is Christine and then this Eric. Both of them, they won Nobel Prize in 1999 for the discovery of this Drosophila wingless gene. So eventually the counterpart of this INT1 gene is this Drosophila wingless gene which they discovered, and they have shown that this gene is very important for, it's a wingless, they called it wingless because the fly, you see this first fly over here, it does not have any wings, or the other fly which has very curtailed wings, or this fly which has only one wing and not the other one. This is your wild type fly which has both the wings. So what these people showed that they say that this wingless gene is very important as a segment polarity gene, and it's involved in the formation of body axis during embryonic development. That is the discovery did. Who did? These two people did. Okay. Anyways, something which is interesting for you guys to remember, this Christine or this woman, she is apparently the first and the only woman from Germany who has won Nobel Prize in science. So maybe for all of you who celebrated Women's Day, you can draw some motivation from her. And it's really, I did not know. I mean, this is, I read on Wikipedia, okay, don't quote me, you can go and double check if after her any German woman has won any Nobel. But it's very inspiring, right? And that to doing developmental biology, hardcore developmental biology, and doing something really cool. So these guys said that this wingless gene is very important. You came too early, yaar. Where in experiment? थोड़ा और डिले हो जाता तो विंगलेस पूरा निकल जाता. So these guys were the first one who said that this gene is very important during the development for the polarity and all. That's how these genes are. So you might have heard about these two pathways, Wnt and hedgehog, or you might have heard about something called as morphogens, no? Morphogens or something, there is a gradient of morphogen happens and all. Both these pathways, they function like that. So there is a gradient of amount of Wnt which is there throughout this anterior posterior development. So when we talk about polarity and development, so when you know, right, where is, if you have like this, what is your posterior, what is your anterior side, this is the typical developmental biologists they study. And we will talk about some very interesting experiment what people did to know about the polarity and all that. So these guys discovered this gene called wingless. These people discovered this locus which is integrin 1. So eventually this WG and this INT came together, and the gene which eventually was discovered called Wnt1, and that's how it became Wnt pathway. Okay, and then they discovered Wnt1 gene eventually. So I also did not know, I learnt it yesterday when I was preparing for the class, but it's very interesting to know the discovery, how it happened. And eventually all of us we know that Wnt signaling pathway is very important when we talk about the development, especially the body axis patterning, or it is very important during the polarity. You go and pick any developmental biology book, any organism which is being development, which undergoing development, this anterior, posterior and all this, one of the very important signaling pathways your Wnt pathway. So that's the discovery of your Wnt pathway, and then they discovered it. Now let's get to the Wnt ligand. So Wnt is the protein, right, which is the ligand over here, and it gets secreted out. So let's see how does it get secreted out. And most of these figures I have taken from one review which was published in 2022 in Nature Molecular Biology Reviews. I have given the link over here at the bottom. You guys can go back and check it. I think it's one of the latest reviews, and I felt it was very detailed information they have given. So you guys can go back and refer it as well. So this is the general scheme of how this Wnt is gonna get synthesized and secreted out. Remember, as we are talking about this particular ligand, a Wnt protein which gets secreted out, and it has certain roles to play. It undergoes some kind of modification, some post-translational modifications when they are synthesized. And eventually, you guys must have heard or read about the secretory pathway, right? You guys know what is the secretory pathway? Science? Yes? What is that? Right. So that's what also do you remember? Anybody else? What else do we remember about the secretory pathway?
Two types of retrograde or
Anterograde. That's what he was telling. Right. Okay. Anterograde means what? Yeah. So from where to where now? Right. From ER to membrane. And retrograde is outer to inside. So when we talk about secretory pathways, you guys remember where does where does transcription take place?
On the ER.
Transcription. Where do transcription? Nucleus. Everybody, have you heard about central dogma, Vaishnav? Yes? What is the central dogma? RNA to protein. Very good. So where does transcription take place? Inside the nucleus. Then what happens? Teja, have you heard about this? Very good. Then what happens?
It's like in eukaryotes, lysine happens inside the nucleus.
Inside the nucleus. Okay. Then?
Then mRNA enters the cytosol,
Mm-hmm.
and then it undergoes translation.
The splicing, I think, happens in the cytosol, in the nucleus. Okay. So RNA gets 3 modifications, right? 5 prime cap, 3 prime polyadenylation, and splicing, and then it comes out of the nucleus. What happens in the cytosol? Translation happens. Now imagine why we called a secretory pathway in yeast Saccharomyces cerevisiae. When people started studying some of these proteins, they started looking at the secretion or the proteins which goes to the plasma membrane or the proteins which get secreted outside the cell. Yeast provided a very good system. What they did, they started doing some random mutagenesis and look at the secretory phenotype, right. If you remove gene X, whether this protein Y goes to the membrane or no, that is the simple assay. Based on that, if you remove this protein A, you say that okay, this protein, if you mutate this protein and this your gene of the other protein of your interest does not go to the membrane, then you call this gene is involved in secretory pathway. That is how that secretory pathway means. Once your mRNA is gets translated, your ribosomes will go and recognize the ATG, then they will start the elongation, and the amino acid tRNAs will bring the correct amino acids, the codon anticodon interaction will happen, and the amino acids will be added. That is how you will start growing the polypeptide chain, right. That is called as a nascent polypeptide chain, because it is just coming out of the ribosome, it has nothing to do with it. And most of the proteins which functions at the plasma membrane, they carry certain modifications. And what is the most common modification they have? Very good, glycosylation. Where does it happen? It happens mostly in Golgi. So the polypeptide, the nascent polypeptide which is just coming out of your ribosome is carrying, there is a signal peptide, no, that is called as signal peptide. That is very important for a molecule, a protein, a polypeptide to go and get into ER. Then there is something called as signal recognition particle. So signal recognition particle goes, recognizes the signal peptide, takes it to the ER membrane. At ER membrane, there is a signal recognition particle receptor present. These two guys talk to each other. Once they talk to each other, there is a channel that is called as Sec61 alpha. That channel gets opened up and this peptide slowly starts entering at the expense of a GTP. Once it starts entering, there is a called as signal peptidase that comes, cleaves the peptide, signal peptide, and then your polypeptide enters the ER lumen. Once it enters the ER lumen, then there is isomerization will happen, PDE1 enzyme comes and disulfide linkages will happen. Once these disulfide linkages will happen, that is when your protein or the polypeptide will start gaining the secondary structure of your protein. Is it clear? This all is happening where? In ER lumen. Like that, if your Wnt, which is a protein which has to get modified, will go to ER first, right, and then these molecules will get packaged into the vesicles and then they enter where? To cis Golgi, right, which is a very dynamic structure. The vesicles are constantly coming and fusing, coming and fusing. There are several models how cis Golgi is being developed. Some people say that vesicles from ER come together, they fuse and then the cis Golgi is formed. Some people think that there is another structure which keeps coming, and then the cis Golgi matures into medial Golgi, medial Golgi matures into the trans Golgi and cis to trans, right, and then trans Golgi is the sorting station. So what happens when these vesicles will come out carrying your protein of your interest, they undergo post translational modifications and all the glycosylation machinery that is present in your Golgi. So from cis to trans, they get modified, get packaged in the vesicle, and then they go to the membrane. Is it clear? Now in this case with your Wnt as well is something similar happening, where this Wnt which is over here, it gets post translationally modified, where it gets palmitoylated, meaning a palmitoyl moiety is added to that, and it is done by O-acyl transferase, which is called porcupine. I do not know, there must be an interesting story why this protein is named porcupine, but I do not know. Maybe
Sir, I think it was because absence of this protein caused the Drosophila to develop spikes on its body.
Spike-like
so.
structure.
That's what I was also thinking, maybe something like that. So there is this hedgehog pathway, no, those they have something, the hedgehog kind of phenotype, it seems. That's how they discovered it. And so this protein is very important now, right? So now you found this, and then it gets essentially acyl CoA is used, porcupine will palmitoylate this Wnt, and then there is wingless. So WLS is a wingless, which is the receptor for this. So this Wnt is bound to this, and then they get attached into these vesicles, as I think Shine was talking on. Aman, you mentioned about the retrograde antrograde vesicles. They are always coated with this COP. You guys remember there are 3 coats, right? COP1, COP2 and clathrine. So COP1, COP2 functions over here. COP2 is for anterograde, COP1 is for retrograde. It's opposite, Vaishnav. Once they go there, and there are so many other 1000 proteins they do, we need not to bother about them. There are snares, T snare, V snare and what not. Let's not worry about it. The only thing is your Wnt gets synthesized in ER, it gets modified, bounds to Wintless, gets to the plasma membrane through Golgi. Now, what people have not understood, that once your this Wnt is out in extracellular space, how does it get separated from your Wintless and all that? There are still people trying to understand, right? That's another theory. Another way what happens is that, so the next thing what happens is that once your Wnt goes outside, your Wintless will deliver the protein outside, the Wnt ligand outside, and the receptor gets endocytosed over here. Now, can you see this? This is whatever you saw over here, this is what is happening. ER, we have Wnt gets binds to Wintless via Golgi, goes to the membrane, right? So this is where it touches the membrane. Now, how this Wintless releases this Wnt in the extracellular space is something that people are still trying to understand, but what we know that it's get released. There are certain models about it that we'll check now after next slide. But the empty receptor now undergoes this whatever endocytosis, once clathrine dependent endocytosis, it comes to the early endosomes, and that's where your retrograde trafficking can takes place, where there is some complex called retromer. Retromer complex will have APS 26, 29 and I think VPS 35. So these are other retromer complex proteins, they come into picture, and there are some sortinexin family proteins. They have these bar domains which can bend the membranes. This is very interesting space actually. I really like how these small tiny vesicles are being trafficked in and out, and why those proteins come, because even in your early endosome, which is such a huge domain, there are some micro domains, and whatever proteins I told you, they are not present throughout the early endosome, they are present only at the certain location, because that's where your Wintless will be there. And from that's the place where they will make a decision whether Wintless has to go to the lysosomes to get degraded, or it should get back to the ER, so that it can go and bind another Wnt molecule and get back to the membrane. That's what is happening over here. So the Wintless, which either there is a retromer dependent recycling, meaning retromer will attach it, once it attaches it comes to the ER, and then it will start going back, or it could get degraded in lysosomes. So essentially what we are seeing is that how your Wnt ligands are being biosynthesized and they are taken in the outside space. Again, going back to this question, how your Wintless releases Wnt, there are certain theories about it, or there are certain models which people have. What is this? There are certain models which people have proposed about it, and then we will quickly talk about them now. So this is slightly busy slide, and there are so much information. We will go step by step. Don't try to look at everything together. Just follow what I say. Then it will become easy for us to understand. Now, there are a couple of models that people have proposed. One, the Wnt which is secreted outside, it will form the Wnt micelles, through their palmitoyl moieties, they will come together and they will kind of a mycelial structure. That could be one mechanism of getting them together. Another mechanism is there is proteins called glypicans. These are glycoproteins, okay? Meaning these are the proteins which are present at the membrane, which have this heparan sulfate and all those molecules are there, no, which are in extracellular space. They are modified, they have glycosylation molecules and they have the ability to go and bind to this Wnt palmitylation moieties. Is it becoming too much or you guys are still following me? Yes, no? No.
Few doubts. So these
Yes.
ABC are different types of liquidation of winds.
Which one?
These ABC, these are different types.
Here.
Yes, sir. A, B, and
ABC are the figures. Maybe I tried to put that small into thing. So, first figure is just telling us about how wind can get secreted outside the cell. It follows this anterograde trafficking or secretory pathway. What second figure takes is what happens to the Wnt receptor, which is windless. Once the windless receptor takes wind along with it to the extracellular space or membrane, it releases out and then it comes back. That is what the second figure is telling you. The third figure is telling you is that how this wind which is secreted outside is going to come together, how it can be released, and what are the molecules which will help this wind ligand to go and bind to its actual receptor, that is frizzled. Is it clear? So, when we talk about wind, the windlass is the receptor for wind to get secreted out. It is not the receptor which is going to do job as such of now. You guys understand? In your receptor, for example, your receptor tyrosine kinases, your RTK HER2 is a receptor, right? That HER2 will be there. EGF is a ligand. That EGF, how it gets secreted, maybe it is a small molecule, maybe it gets modified and secreted. In case of wind, that is not that easy. Even to carry this ligand from the cell to the extracellular space, there is another receptor which does this job. Is it clear now? So, unlike your EGF or any other cytokines, these molecules can be released in outside space. Your Wnt can also be released, but it cannot be just released like that. That is why you need a separate molecule. It is kind of chaperone, it is kind of an entity which is going to help this molecule to go outside. That is the job of windlass over here. But actual receptor which plays important role in the signaling, the quote unquote your receptor tyrosine kinase equivalent moiety over here or a GPCR equivalent moiety over here is your frizzled receptor. It is not the windlass. Is it clear everybody? Good. So, in this case what is happening at early endosomes, now just pay little bit attention with this. So, one way how your wind can get out from this windlass in extracellular space is by formation of wind micelles like this, okay? That is one way. Again, there might be some experimental evidence to that. I did not go back to the paper to find out the experimental result, but these are the some models which are there currently people working on, okay? I am not saying this is the model, this is one of the model which is proposed, okay? It is not established model, but this is one of the way people think. Another thing is, you guys remember I was telling you about the when your EGF receptor complex comes to the early endosomes, based on the ligand what it binds, it can be either get degraded or recycled, right? So, the pH of your early endosome decides something. So, in this case what they say is that whenever your windlass and Wnt is at the early endosomes, okay, what happens that there is a possibility that because of the low pH your windlass and Wnt can get separated from your windlass which is the receptor, and then there is another molecule called as this DLP like glypican, which is another glycoprotein which functions at the membrane, plasma membrane. And you can see the way this structure is. I do not know how clearly you can see that if this is your Wnt molecule and this is its palmitoylated moiety, you can see that there is this black thread kind of thing which is there along with this, and this is your glycopan, right, where they are trying to shield this moiety That is how they are chaperoning it. So, because it is a lipid moiety, if it gets, it will start aggregating everywhere, right. That is why these molecules which are glycoproteins, they are kind of shielding this moiety over here, and rest of the structure is accessible, and then this vesicle will go out and they will release it. Is it clear? Where this DLP domain containing proteins like glypicans are there, and they have some modifications, carbohydrate modifications on them, because they are the carbohydrate glycoproteins. Is it clear? Teja? Yes? No? Gaurav, did you understand? Everybody understood this? Rishika, did you get it? No? You want me to repeat?
Sir, after you said that the windlass and the path is getting separated in the early endosome, then what is happening?
So, when they get separated over here, right. So, now your Wnt ligand is free, this orange color moiety. So, there is another protein called this glypicans. Okay. They have certain structures through which they go and shield the lipid moiety, because your Wnt gets modified with lipid, right, palmitylation happens. So, what is more important is you go and shield this, and that is what has happened. If you look at this, if you look at this black thread kind of structure here, there is some knot kind of thing over here, right. That is when your this orange color tiny palmitoyl moiety is there, and rest these kind of. So, here in this structure, this is how your glypican structure looks like, right, where this is dark black thread and then this is the structure. So, these are these are glycoproteins. What do we understand by glycoproteins? They have modified with carbohydrates and they have the proteins, right. So, they will have these there is something called as heparan sulfate, which is present in extracellular space. These proteins do carry those kind of modifications, and this black thread like structure what you see at the top, this is where they actually hold the lipid moiety of your Wnt, and then they shield the Wnt ligand, and that is how these molecules will be taken in the vesicle and then they are secreted outside. Now, is it clear? And now, if you look at it this at the membrane, there is another question mark and dotted arrow over here, because we do not know from this molecule how do we transfer it to the free result. That is again another question, how it is happening, maybe people are still understanding. Yes.
It is not in the same cell, right?
Same cells. Yes, yes. So it is possible. So, this the way they have shown it, if it is what is that called as autocrine signaling, the cell which will secrete the molecule, they carry the receptor. It is possible, or if they can do juxtacrine signaling, where the next cell also can do it. Here they try to show next result, but it is possible because next to each other there is another receptor that can go and catch it up. That I will have to check. And then there are exosome dependent pathways, which I do not know whether we should really start extending ourselves into that, and then there are cytoneme structures where they are present. So, there are multiple mechanisms. The only thing what you guys should really remember is that Wnt ligand, or the ligand of the whole pathway is Wnt, right, that it is a protein, but it is lipidated, it has palmitylation, and who does it? There is a transferase called porcupine, and once they get it, they follow the secretory pathway to get out of the cell. How does it get out of the cell? Once via use of Wnt-less receptor. But how this Wnt-less receptor releases into the extracellular space, there are certain models about it. One of the model is Wnt micelle formation or roll of glypicans, where they shield them and then take them outside. Is it clear? Yes? No? Good.
One of the model is Wnt micelle.
Right. Second is this model. This is the second model. It is not the same model. So, this figure summarizes all the models, like this micelle formation model, or glypican model, or exosomes model, or cytoneme model. There are 4 models which are summarized in the same picture. Exocytosis, have you guys heard about exocytosis any time? No? How many of you have heard? Shivam, what do we know?
So exocytosis means how the substance is moving out of the cell.
Right. Anybody else? Yeah, you want to add more?
So they form something like the package is formed and then it is get like connected to plasma membrane. Modification is occurring through which it is moving out of the cell.
Right. Anybody else want to talk about it? Nobody is working on extracellular vesicles, exocytosis, blah blah blah. No one? Teja? Exocytosis, yes. So essentially what is happening is that, so we talked about these early endosomes, right? Here. Now, at early endosomes, you have your Wnt ligand containing, Wnt is there, this is this tiny moiety is there, let us say this is your, it's pretty bad but it's okay. Like this your Wnt is there in early endosomes. Now, there are several mechanisms how this can go outside, right? One with through Wintless, Wintless के साथ इधर आ गया, यहाँ से glypicans can take them, those those glycoproteins. The another way is what happens is that the canonical endolysosomal trafficking pathway, we have early endosomes, right? These early endosomes, they are very dynamic structures, they undergo homotypic fission and fusion and then they start growing, forming late endosomes. Okay. What do we understand by this? Meaning there are different, when this, there are so many early endosomes like this, right? They undergo, they come together, they fuse it and it's like two bubbles coming together forming a big bubble. Then three bubbles coming forming a big bubble. Then they reach a big structure, they form a bigger structure. Okay? And then what happens in this bigger structure, which they call late endosomes, which mature into formation of multivesicular bodies, what happens in this big bubble, there will be small bubbles. Meaning from these early endosomes, they form a structure which will be something like this, which is called as big vesicle. Inside this there will be small vesicles. Okay? And in this each vesicle would carry your cargo. Like in this case your, maybe your, what is that called as, Wnt ligand. Did you follow? So here Wnt ligand was present. What happened? Your, how did I draw that structure? This glypican or something comes, packages, right? Now what happens? Maybe, okay, I will put it here. This is fine. Now what happens? Let's say your, this is your glypican which comes and attaches to this side, right? Now what will happen? There will be some proteins, they will come and then they will form a vesicle like this. Okay? What would happen? This vesicle which is at the early endosome, it can come out of early endosome and then this molecule will be packaged as a single vesicle and goes to the membrane. Is it clear now from this picture? Sometimes what happens, your early endosomes, when they have these small vesicles, they will fuse together forming a bigger vesicle, and in this big vesicle we will have these small vesicles, each vesicle will carry one Wnt ligand. Okay? It is like bulk secretion is going to happen, as Teja was telling. You guys have heard about insulin secretion, insulin containing excuse me, insulin containing granules or something which happens, mostly digestive enzymes will be there in your cells, right? They are released in bulk. If you need bulk amount, maybe this method would be followed, where you have so many tiny vesicles in a big vesicle, and as Shivam said this vesicle can go and fuse with the plasma membrane and release the content outside. That is where exocytosis is happening. Is it clear? That could be another method. Yes.
Are glypicans required for the wing to get recognized by frizzle?
So that's the question that we do not know. That's why you see that there is a dotted arrow and a question mark on that.
Because even when it is exocytosed and maybe it is doing the juxtacrine signaling, glypicans are released along with the wing.
It is possible that they hold the ligand like this so that the receptor can bind it. But I do not know. I have not read a paper where they say that if you deplete glypican, see it, I do not know the recent literature, but you can think about it, right. If this molecule is important to take the ligand itself outside, if you do not have glypican, whatever effect you are seeing, it could be because of two reasons. One reason is because it cannot get secreted out, and the another reason we cannot differentiate unless your glypican is present outside and then there only it is doing job. Where you remove only the surface exposed glypican and you see in that case what happens, whether frizzle can bind to this wind or no. Did you understand what I am trying to say, right? So, I do not know the recent literature. It is possible that it is required because this the way they have shown this arrow, and this is one of the recent reviews. I would think that maybe it helps in the concentration of these winds next to the frizzle receptor, so that receptor can recognize it. Like what happened with your beta TGF beta signaling, where R1 it does not have catalytic activity, but it can sequester the ligand. It has the ability to bind to the ligand and maybe it presents it to the next receptor. So, this is possible, but I do not know, I cannot tell you that it happens because I have not seen an experiment where they have done it, and maybe that is why they have kept the arrow. Yes, you have. No, this picture does not say that they require glypicans, they just through your windlass. Yes, that is an independent pathway. Yes, it is a different pathway. No, so that's exactly why I was trying to tell you guys. So there are multiple models how people have studied. So maybe somebody studied and they found that this new molecule is required for secretion. Somebody other molecule people find that this is the receptor which is required for secretion. We do not know. Maybe in the cell all the pathways operate simultaneously, because you can understand, right, this and something which is very important for us to remember, these are morphogens, right, and these are not long range morphogens, they are short range, meaning once they secrete, only in that range they are going to do secretion, and that is when your glypicans become important, because your juxtacrine signaling might become more relevant. You understand what I am saying, right? There is a gradient of wind which will happen, that I will show you now. Meaning, if let us say I am a cell which is secreting wind, right, or let us say if I am a cell which is secreting wind, I can secrete and only Vaishnav can get the wind ligand, not you, because it is a short range. So, if Vaishnav has to signal or it has to get activated, maybe here there is a glypican which is trying to hold it and present it to him, but I cannot reach to you. So, that is why there is a short range gradient which is formed with these wind pathway, unlike maybe there are other pathways where there is a long range morphogen gradient can be formed. So, that is when they might become important. So, anyways, apart from biogenesis, this is what you guys already know, what is the overview of the pathway when the pathway is inactive. Remember, there are so many names to if you guys want to remember. I think at this stage you should remember these names, and you all are biology students. So, I mean sometimes for those minor students I give them option that even if you do not know every name, it is fine, but these are 5-6 molecules. Already you guys remember so many names. So, frizzled is the receptor which is present at the membrane, and LRP5 or 6 is a co-receptor kind of. This molecule has to be there for wind pathway to activate. So, GSK3 beta is a kinase, casein kinase 1 is another kinase. Okay. the most important thing what you have to remember is that your beta catenin is the transcription factor. Beta catenin is also present at the plasma membrane, but the beta catenin which is free in cytosol, that is what will get translocated to the nucleus. So, then the pathway is inactivated, the whole system is running behind to inhibit beta catenin function and avoid its entry into the nucleus. How do they achieve it? They, first of all, there is this protein called axin 1 that acts as a scaffold, it is a scaffolding protein that goes and binds to beta catenin. Then it will recruit this GSK3 beta and casein kinase 1, they will phosphorylate this beta catenin, and this phosphorylated beta catenin becomes an excellent substrate for an ubiquitin E3 ligase called TRCP. So, axin 1 is the protein which is going to hold your beta catenin at one place in cytosol. So, that beta GSK3 beta casein kinase 1 come and phosphorylate. Once it gets phosphorylated, your beta TRCP will come, ubiquitinate this one and then degrade in where? जोर से proteasome. In cytosol only everything is happening, but it will get degraded in proteasome. So, which means that these chains will be जोर से K48 UBK48 chains will happen, right, because it is undergoing proteasomal degradation. And when your pathway is on, what happens? Wind goes and binds to a Frizzle receptor, and there is this protein called dishevelled, which most of you talked about. Still people I think are trying to understand its actual molecular function, but maybe it acts as a scaffold or some initiator where it sequesters your axin to the membrane, and this axin, APC, GSK3 beta, casein kinase 1, everything will be sequestered at the membrane, so that your beta catenin is happy in the cytosol. Then it enters the nucleus and then it goes and binds to the TCF and then do the transcription of the Wnt signal, Wnt related genes. Pathway is very straightforward, but the whole mechanism is dependent on the ubiquitination machinery, and then eventually there might be other molecules which regulators people might have identified. Is it clear? Everybody? Pathway is very straightforward, right? So, now let us see what I was talking to tell you about the Wnt. And do you guys know planaria? Everybody has seen, heard planaria. It can regenerate itself. How? Because what did he say? Because they have, say you are right, maybe stem cells. You guys never heard about how planaria regenerates itself? No? Good. Then my whatever video I will show that you guys can, you won't sleep in it. So, they have adult stem cells present in them. So, even if you cut planaria into middle, it can still form a new head or it can form new tail, or even if you have only this space, this piece, still it can form a new planaria. You understand what I mean? So, that is the ability it has, but the formation of head at which end, the formation of tail at which end, or the marking of posterior and anterior segments is whole determined by your Wnt signaling. And if you see in this figure over here, this posterior is this tail, anterior is its head, okay? And you can see this magenta kind of color gradation you see, right, which is much more over here, whereas it is decreasing down, which means that your Wnt is more active at this end and this reduces that end. So, which means that your beta catenin is going to function to this end, so that it is required to form the tail, and if this way if you go, then it is the less Wnt is required to form the head and more Wnt is required to form the tail. Is it clear from this picture? Do you guys understand? That is the gradient of morphogen which has happened. Do you guys understand how interesting it is? And that is one of the experiment what they did to understand the, and then you can understand. So, which means, I mean in this case they have used another negative regulator called NOTUM mRNA, which is required to form the head. So, if you inhibit beta catenin, two heads are formed. If you inhibit NOTUM, two tails are formed, and if you inhibit both of them, which still two heads are forming. So, they have put it in perspective that whenever there is a signaling pathway happening, wind gets secreted, it will form beta catenin. When beta catenin is there, it will form tail. When beta catenin is not there, it will form head. But if you have more and more beta catenin, then it will still form, it will form only tail and no head. So, this is how the whole dynamics of wind signaling is